We've all heard of the hygiene hypothesis, but what does modern immunology actually say about the link between environment and allergy? Well, a new 2026 Nature study offers the clearest mechanistic evidence yet. Natural immune stimulation can block allergic sensitization at its source. If you're a student, researcher, or clinician trying to understand how early life exposures shape IgE, IgG, and anaphylaxis risk, this is the breakdown you've been waiting for. Hi everyone, I'm Dr. Susan Heavey, Associate Professor at UCL, and this is Further Reading. Today we're looking at a translational immunology paper published in Nature in 2026. It's called Environmentally Driven Immune Imprinting Protects Against Allergy. It's a completely open access article. The link is in the description and I'm going to discuss it panel by panel in the next chapter of the video, but I'll start with a quick overview and summary. So let's start with why this topic and paper matters. Across the world, allergic diseases are rising sharply. Genetics alone cannot explain the increase. Epidemiology points strongly towards environmental drivers, but the field has lacked a clear mechanistic explanation for how early life or cumulative exposures shape long-term allergic risk. This paper addresses that gap using a mouse model. The core idea is deceptively simple. Mice raised in a naturally diverse immunostimulatory environment, as they call them here, pet shop mice, develop broad cross-reactive immune memory that protects them from allergic sensitization and anaphylaxis. The authors compare these pet shop mice, who have wild-type exposures, diverse microbes, environmental antigens, against SPF mice. That's specific pathogen-free mice, or lab mice. They're extremely clean. 5 key findings are, firstly, that pet shop mice are protected from allergy across multiple sensitization routes. Secondly, they carry pre-existing cross-reactive IgG and memory T cells to antigens that they've never even seen. Thirdly, there's a perinatal window where allergic sensitization is possible. After that, environmental imprinting closes the window. Fourthly, type 1 based or tolerogenic exposures to related antigens can protect against allergy. And finally, this mechanism includes IgG-IgE balance, cross-reactive memory, and contextual imprinting. I'm going to go through a figure-by-figure walkthrough next. This will be just the main paper. This is, of course, a Nature paper, so there are approximately 20 million supplementary figures. If you want to discuss it in a deep level, you can join us on Substack. But for panel by panel of the main figures, stick around for the next chapter. Okay, let's start with figure 1. In panel A, we see the experimental design. Mice were sensitized to ovalbumin, or COVA, on days 0 and 7 via skin, lung, or intestine using type 2 driving adjuvants, and challenged systematically with COVA on day 14 to assess anaphylaxis. This panel simply shows the timeline and routes used to induce allergic sensitization. Moving on to panels B to D, this shows intestinal, lung, and skin sensitization. The left plots, SPF mice show a sharp drop in core temperature after the COVA challenge, while pet shop mice lose little or no temperature, indicating strong natural protection from anaphylaxis. In the middle of plots, we see maximal temperature loss, confirming that this protection is across all routes. And on the right plots, pet shop mice produce less IgE after intestinal and lung sensitization, but equal or higher IgE after skin sensitization, showing that IgE levels alone do not explain their protection. In panel E, we see the setup for the passive systemic anaphylaxis. This schematic shows how IgE-mediated passive anaphylaxis was induced using the anti-DNP IgE, followed by DNP-HSA challenge, bypassing the normal sensitization phase. In panels F and G, we see passive IgE-mediated anaphylaxis. SPF and pet shop mice show similar drops in temperature when anaphylaxis is triggered directly through transferred IgE, indicating normal mast cell-mediated effector function in both groups. This demonstrates that protection in pet shop mice does not arise from defective mast cell activation. In panels H to J, we see direct mast cell and mediator activation. When mice receive compound 4880, that's mast cell degranulator, histamine or PAF, both SPF and pet shop mice respond similarly. These results show that downstream vascular and mediator-driven pathways are intact, confirming the protection occurs upstream of mast cell activation at the sensitization or IgE engagement stage. So your take-home message for this figure is that pet shop mice are protected from allergic anaphylaxis because their prior environmental immune experience prevents allergic sensitization, not because their mast cells or downstream anaphylactic pathways are defective. In Figure 2a, we see that pet shop mice already have COVA-reactive IgG despite no exposure, showing pre-existing cross-reactive antibodies. In panel B, we see that epitope profiling reveals broad antibody binding to many COVA peptides in pet shop mice, but not SPF mice, indicating environment-driven cross-reactivity. In panel C, ELISpot images show pet shop splenocytes produce interferon gamma when stimulated with COVA or KLH, unlike SPF mice. In panels D to F, quantified interferon gamma spots confirm strong baseline and antigen-induced type 1 memory in pet shop mice, absent in SPF mice. In panel G, flow cytometry identifies COVA-reactive CD4-positive memory T cells in pet shop mice, but not SPF controls. Finally, in panel H, pet shop mice show significantly more COVA-reactive interferon gamma-positive CD4-positive T cells, confirming widespread pre-existing memory. Your take-home message for this figure is that pet shop mice possess broad environmentally generated antibody and T cell memory to novel antigens, meaning they are not immunologically naive the way that SPF mice are. Figure 3 overviews what I hesitate to call a super cute fostering experiment where the pet shop pups were raised by SPF dams and they lost their environmental exposures. In panels B and C, we see that fostered pet shop mice become susceptible to anaphylaxis and produce less IgG2, meaning protection depends on early life environment, not genetics. In panel D, we see the design comparing perinatal P7 versus adult sensitization in pet shop mice. And in panels E and F, we see that pet shop mice sensitized as neonates show strong anaphylaxis and type 2 biased IgE, IgG1, whereas adult sensitized pet shop mice remain protected and generate mixed IgG responses. Your take-home message for Figure 3 is that allergy susceptibility exists only during an early life window in pet shop mice. Environmental immune experience closes that window and shifts them into a long-term non-allergic set point. Okay, on to Figure 4. In panel A, IgG from COVA-sensitized SPF mice binds not only COVA but also OVA orthologues, that's Q ova, D ova, and BT ova, showing broad cross-reactivity across avian ovalbumins. In panel B, we see an experimental design where mice first receive a type 1 biased immunization, that's OVA or orthologues and CFA, then undergo standard allergic sensitization with COVA or alum. In panel C to D, mice pre-immunized with C ova, Q ova, or D ova are protected from anaphylaxis and show higher COVA-reactive IgG, while CFA-only controls remain fully anaphylactic. In panel E, higher IgG to IgE ratios strongly correlate with protection from anaphylaxis. In panel F, we see the post-sensitization CFA immunization design, giving a type 1 signal after mice that are already allergic. And in panels G to H, even after allergy is established, exposure to COVA or Q ova in a type 1 context reverses anaphylaxis susceptibility by boosting IgG without increasing IgE. Your take-home message for Figure 4 is that prior or even post-allergic type 1 imprinting with cross-reactive antigen boosts IgG and protects against anaphylaxis, showing that adaptive imprinting across antigenic space can prevent or reverse allergic sensitization. Okay, on to Figure 5. In panel A, we see the oral tolerance model. Mice drink COVA and water before sensitization to induce a tolerogenic, non-inflammatory exposure. In panels B to D, oral COVA tolerance reduces anaphylaxis and lowers COVA-reactive IgE, confirming that tolerogenic exposure suppresses allergic sensitization. In panels E through J, oral tolerance to COVA also protects against sensitization to OVA orthologues, that's Q ova, D ova, and BT ova, decreasing IgE and anaphylaxis across a wide identity range. This shows cross-tolerance spreads across antigenic space. In panel K, we see that the degree of protection correlates with the sequence similarity between COVA and each orthologue. In panel I, we see the diet experiment design. Mice raised on chow, which contains soy versus soy-free diets to test real-world complex antigen exposure. And in panels M through S, soy in the diet induces tolerance not only to soy but also cross-tolerance to other legumes such as pea and peanut, reducing IgE and anaphylaxis. Soy-free mice react more strongly. This demonstrates dietary proteins generate broad, cross-reactive oral tolerance in complex natural antigen mixtures. Your take-home message for Figure 5 is that allergenic exposure, whether to a single protein or a complex dietary mixture, creates broad cross-tolerance that suppresses IgE responses and protects against anaphylaxis across diverse related antigens. Let's take a minute to talk about the mice used in this work. So they used SPF for specific pathogen-free mice, and these are the standard for controlled immunology experiments. They represent one end of the spectrum, and they are the perfect choice here. Pep shop mice represent the opposite extreme. They've been exposed to all of the antigens, all of the mess, all of the dirt. Again, the right choice here. I wanted to look into whether other options could have been included. Generally, as someone who works with different kinds of models in the lab, I usually like to see multiple models used to just kind of validate and give me a little bit more confidence in the findings. And I guess having looked into this, they could have tried wild-caught mice. It's not very practical, and I think pet shop mice are a pretty good, not even compromise, but a pretty good logistic decision here. They could have also used rewilded lab mice, which is something that other groups have done, where they take mice sort of like the SPF ones and then just create a sort of artificial or as close to nature version as they can, where these mice are exposed to lots of different antigens. Could have been done, but not that practical. They could have also used defined microbiota mice, but again, it's maybe not the best choice. It's probably too specific, not all-encompassing enough. So overall, I got to say they pretty much made the right decision here, unsurprisingly, given it's a Nature paper. And it's worth noting that they did do a few secondary exposure paradigms here. So they co-housed the SPF mice. They also did those really nice fostering experiments and the adult versus perinatal sensitization. So really, by looking at both ends of the spectrum with the SPF versus the pet shop mice, I think they had a really nice experimental design here, and these mice pretty nicely represent the human scenarios they're trying to look into. As with all mouse work, we do have the usual caveat that mice are not humans. Their biology is very different, but this is not the kind of work that we can necessarily do in humans, and it's really pretty nice what they've done here. So my compliments to the authors. Okay, before we wrap up, I wanted to do a little overview for students specifically, because this paper is really a goldmine for boosting your allergy and immunology exam answers, as well as any coursework or presentations that you're going to be doing in relation to this topic. So firstly, this is an update to the hygiene hypothesis. Most students will say allergies are rising because we're quote unquote too clean, but you can go further. Environmental immune experience creates cross-reactive IgG and memory T cells that block allergic sensitization. One sentence like that immediately raises the level of your answer. You can also explain why infants get allergies more easily. So instead of saying the immune system is immature, you can add there's a developmental window where sensitization is possible. After that, accumulated immune experience shifts the system away from allergy. Markers love that level of insight. You can also elevate sensitization pathway answers. So everyone writes about IL-4, IgE, and mast cells, but you can add sensitization isn't guaranteed. Environmental imprinting can stop Th2 polarization entirely, even with strong adjuvants. This shows a deeper mechanistic understanding. You can also improve immunotherapy answers. So if you're asked about allergen immunotherapy, you can add that protection correlates with the IgG-IgE ratio, and that cross-reactive IgG can intercept allergen before it reaches IgE. So that ties textbook content to current research. You can even use it in microbiome or environment essays. So this paper lets you say that environmental exposure doesn't just shape the microbiome, it creates cross-reactive immunity across antigenic space, even between food proteins like soy, pea, and peanuts. And we were just chatting about mice, and you can do this too. You can add it in when you're discussing mouse model types. You can say that SPF mice resemble immunologically naive neonates, whereas pet shop mice resemble environmentally experienced adults. That's a sophisticated point that a lot of students might not make. So if you drop just one or two of these ideas into an exam-style answer, you'll stand out immediately as someone who understands not just the pathways, but the content and modern mechanisms of allergy biology. It's a simple upgrade that markers really do notice, especially if you can actually cite it as this specific paper, because it being Nature and 2026 shows that you're up to date on top-level literature and just gives a level of prestige to your answer. Enjoy. Okay, that's it for this episode of Further Reading. There's plenty more videos available if you want them on the channel, and if you prefer a written version, you can check out my newsletter via Substack. There's links in the description, the comments, the bio, all of the places. Enjoy. If you made it this far into the video, well done. Good luck.